All right. It looks like we're at the start of the hour, so we're going to go ahead and get started. So first of all, hi and welcome everyone to the webinar. We're really excited to have you here, and thank you so much for taking the time to join us today. My name is Kayla Ripple, and I'm a principal associate with the Lenfest Ocean Program. So for those of you who may not know us, the Lenfest Ocean Program is a grant-making program, and we fund ocean and coastal research projects to address the needs that are facing decision makers and stakeholders. You can learn more about us and the projects we fund at lenfestocean.org. And while you're there, you can also sign up for our newsletter to receive updates about projects like this one and others as they move forward. If you're on Twitter, be sure to follow us at LenfestOcean. In fact, we'll be live tweeting this webinar today using the hashtag LOPWebinar. So feel free to use it too if you'd like to engage with us there. Today is the fourth webinar in a monthly series that highlights projects from our Climate-Resilient Fisheries grant portfolio. In this portfolio, we're supporting projects where scientists are working with managers and stakeholders all over the US and abroad to better understand how climate change will impact fisheries, and then help fisheries managers and communities develop solutions to foster climate-ready fisheries. So this includes a range of topics such as predicting range shifts of economically important fish stocks, understanding the climate vulnerability of coastal communities, and providing guidance on adaptive management for fisheries, among other things. So if you're interested in learning more about those projects, my colleague Sarah Close will drop a link to the landing page for all those projects in the chat. On this page, you can also view and sign up for upcoming webinars in the series. These occur on the last Wednesday of each month. We've currently scheduled these through June, and we aim to continue scheduling through the remainder of 2024. In fact, our next webinar will be on Wednesday, May 29th at 2 p.m. Eastern, 11 a.m. Pacific time on quantifying relationships of northern fur seals, pollock, and climate change in Alaska. And Sarah's dropped the registration link for that in the chat as well. But today, we're excited to have joining us Dr. Stephanie Green and Miram Gleiber with the University of Alberta. In this project, Stephanie, Miram, and others on the research team have been working with partners at other universities, NOAA Fisheries Science Centers, and elsewhere to develop trait-based methods to predict predator-prey interactions in changing ocean systems. So this project examines historically variable feeding relationships between albacore tuna and the pelagic species they consume in the California current systems as a case study. And Sarah just dropped a link to their project page in the chat, as well as a resources page that includes current publications and other media that has come out around this project. In fact, we also had a new guest blog come out today written by Miram and Stephanie. It literally just went online today, and we're dropping the link there in the chat as well. This talks about some of the results and the importance of collaboration to inform new management tools. So before we get started, just a few logistical things. To prevent any feedback or echoes, all attendees are muted, but we do want to hear from you. So please feel free to drop your comments or other resources you think would be useful to share with others in the chat box during the webinar. And then if you have questions for the speakers, please submit those in the Q&A box. I'll be keeping track of these through the webinar, and at the end, I'll read the questions aloud for the researchers to answer. If we don't get to your question, or if you have follow-up questions, please feel free to reach out to us or the research team after the webinar. We're very excited and happy to follow up with you. And lastly, this webinar is being recorded. We'll distribute the link broadly afterwards, so please feel free to share this with others. And again, follow up with the research team should you have any other questions. And with that, I'll stop sharing my screen and turn it over to Miram to start sharing her screen. And Steph, I'll turn it over to you.
Thanks very much, Kayla, and thank you to the whole Lenfest team for not only supporting this project, but continuing to be a great resource and set of partners and thinking about how we have opportunities to share about this work. Good afternoon, everyone. My name is Stephanie Green. I'm an associate professor at the University of Alberta, and I've had the privilege to be working on this project for the last many years. And it's our pleasure today to tell you about some of the findings and also the background and motivation for the work that we've been doing, thinking about how we might suffuse predictions of how species are going to shift, particularly those that are important for fisheries at the coast, with information on predator-prey relationships. Next slide. I want to start by acknowledging the huge set of partners and collaborators that have made this work possible. We have been working over the last many years with scientists from both the University of Alberta, University of California, San Diego, Stanford University, Oregon State University, University of California, Santa Cruz, as well as NOAA Northwest and Southwest Fisheries Science Centers and various groups within those agencies. And it has been an incredible learning journey and experience. This work draws on both the conceptual input and ideas of the people listed on the screen and also others, as well as data sets and a rich legacy of data that have been collected in the California Current on the west coast of North America. So thank you to everyone here. And the work we're going to share today is as a result of this collective effort. Next slide. And a key interest of this set of collaborators and colleagues, as well as I imagine everyone that's joining in on this webinar today, as well as the Lenfest Ocean Program, is understanding how ocean ecosystems are going to respond to major threats like climate change, and what that is ultimately going to mean for communities, economies, livelihoods, and cultures that rely on those ocean systems. This is a figure from a study that really, I think, to me, helps to encapsulate the scale and the complexity of the problem. It's a global synthesis of how marine species may be redistributed under climate warming scenarios, with the colors here showing either the loss or the gain of species in terms of diversity across latitudes. And as we can see, when we're looking at sort of how climate change is likely to shift the fundamental environments where ocean species can persist and thrive, we see that towards the equator. We are projected and we're observing that will likely lose species, have declines in diversity. And as we move to more temperate and northern and southern latitudes, we'll see that species may begin to move into areas. But the fundamental message of synthesis work like this and observations on the ground is that ocean systems are changing, and they're changing in complex ways where we now have whole new sets of species interacting with one another. And that is really changing the field of how humans are interacting with the ocean as well. Next slide. So we know that many of the species that are in our coastal systems are involved in economically important fisheries for coastal nations. And this is some synthesis work showing the implications of climate change for the management of species that are involved in fisheries with a transboundary nature. In particular, when I say transboundary, I mean a harvested species population that is accessed by fishing fleets from multiple countries or multiple jurisdictions. Essentially, that stock being shared among the economic interests of many coastal communities and nations. And what we know from synthesis work as well is that these transboundary shifts are going to be pervasive and are already occurring. In particular, the synthesis estimated that by 2030, so just over 5 short years from now, it's very likely that almost a quarter of transboundary stocks are likely to shift in their distribution across jurisdictional lines, and that the vast majority of jurisdictions are going to experience shifts in the transboundary nature of their shared interest in fisheries. And so climate impacts are pervasive globally, but they also are likely to manifest as a changing landscape of economic opportunities and also challenges around diplomacy and policymaking at both a national, regional, and international level. Next slide. The region that we're going to focus on today that is a perfect test case for this conundrum of how climate change is affecting transboundary fisheries is in the Northeast Pacific Ocean, where the many hundreds of species that occupy coastal and pelagic environments are projected and in many cases already shifting their ranges and abundances due to climate shifts. In particular, this is just a graphic that's showing the direction and magnitude of shifts that were estimated for over 300 species of marine taxa in the Northeast Pacific. And importantly, the direction, magnitude, and certainty of shifts varies across these species, such that it's not all happening concordantly with one another. And as a result, what we're seeing is that communities essentially are being reassembled, ecological communities with new members as well as relative abundances. Many of the species in this system are also involved in important coastal and also high seas fisheries. And so I want to take you into a little bit of a deeper dive into one of those target species that we're going to focus on for the large part of the remainder of the talk, because it is a perfect illustration of both the potential implications of species range shifts in our coastal systems, what it means for fisheries, and also the possibility of using new scientific approaches to understand what might unfold. Next slide. So I want to introduce you to the albacore tuna. It is a highly migratory, beautiful, silvered, streamlined ocean predator. And in the Pacific Ocean, albacore tuna make large migrations around the ocean basin where they're seeking out key environments and prey resources. So on the right side of the slide here are some tagging data, tracking data that show the movement of albacore tuna in the Northeast Pacific. And fish migrate around the ocean basin, but they actually come to the coast of North America in search of prey resources in summer months as large juveniles. And there is a keen interest in understanding where and when and to what extent those fish will hit the coast, because there they are intercepted in important coastal fisheries, both by Canada, the US, and Mexico. Next slide. In the Northeast Pacific, the albacore fishery is a troll fleet using hook and line. And as a result of that harvest method, there tends to be fairly low bycatch for this species. The population characteristics and also potential for low bycatch of this species mean that currently it is listed as an MSC certified fishery. And crucially, it's open access. So there's no limited entry in terms of the way that licenses to harvest albacore are provided. And that allows them to also be targeted by fishers that are accessing other resources at the coast. So it's emerging as a very important resource in coastal communities in the US, and as I said, is also accessed by fishers in Canada and also Mexico. Interestingly, and important for transboundary fisheries management and science, is that access to albacore on the west coast of North America and in the Eastern Pacific between fishers in Canada and the US is managed by a treaty between the two nations that sets out the parameters for reciprocal fishing in coastal waters off of each nation. The most recent version of the treaty expired in 2022, and unfortunately, there's been no consensus in negotiations to renew that treaty, which means that in the last few years, there's been no reciprocal fishing of each fleet in the other nation's coastal waters. And so understanding what is driving the distribution of albacore and how we might understand what access that's equitable looks like between the US and Canada is really important at this moment in time to allow that treaty to go forward. And crucially, all of this is happening, as we mentioned, on a backdrop of climate change. Albacore are existing in a system where environmental oscillations that have long existed historically are intensifying and changing in their nature. And so there's a keen need to understand what those climate shifts will mean for predators like albacore, their distribution and abundance, and ultimately then how fisheries for them might proceed in future. Next slide. In fact, we know that fishing effort for albacore tuna has fluctuated in the past and is, of course, likely to fluctuate going forward, likely as a result of the environmental conditions and other parameters that albacore facing, as well as the conditions for communities of fishers that harvest this species. So these are some data from a study led by Tim Frawley that worked with fishers to understand how fishing efforts for the albacore fleet was distributed over time. And through that collaborative study, what they observed is that through the past decades, there have been large shifts, both in the latitudinal distribution of the fishery, as well as from onshore to offshore, with centers of fishing shifting both off and onshore, as well as intensifying north and south. And so this is a fishery that in past decades has been variable and is likely to have a changing nature as potentially the distribution of albacore changes. But a key question is, of course, to what extent are these shifts driven by the distribution of albacore themselves, and how might we better understand what the distribution of albacore is going to be to help inform and guide what might be possible in terms of accessing this species. Next slide. These next few slides are from our colleague Barb Muehling, who has been studying the distribution of albacore tuna as well as many other species in the California current large marine ecosystem. And the animation here is sea surface temperature across the North Pacific over time. And the moving dots that you can see are actually tagged albacore that are migrating within the environment. And as you can see, albacore tuna tend to be following bands of favorable environmental conditions, in particular temperature. They have a lifestyle that requires that they closely track environmental conditions and fronts that facilitate their high energy demand lifestyle, but also there's quite a lot of variation. Next slide. So when Barb and colleagues have further dug into understanding what predicts the distribution of albacore tuna, they have been able to look at potential distribution in terms of predicted catch per unit effort in relation to ocean conditions. And so the graph on the left is species distribution model output generated from logbook data of albacore landings in relation to some key environmental predictors, in particular sea surface temperature and surface chlorophyll. And as you can see from the species distribution model, it predicts that there might be high catch per unit effort under a particular range of wide range of temperatures, as well as chlorophyll conditions and an interaction band between the two. And what Barb and her colleagues were able to do is also overlay observations. So to sort of have a little bit of a validation for this model to look at where, in particular, tagged albacore have persisted in the system in relation to these predicted values. And as you can see, on the right is the circles of tagged albacore in relation to those species distribution model outputs. As you can see, there is some concordance with these key environmental temperature and chlorophyll values, but there is also a lot of variation. And so as we think about predicting the distribution of a predator like albacore, understanding their relationship with abiotic environmental conditions like temperature and chlorophyll and others does certainly get us so far, but it's likely that there's other conditions in the system, in particular how their prey are responding to the environment and distributed, that is also modulating the distribution of top predators such as albacore. And so one of the key pursuits of understanding how we can predict distribution and abundance for predators such as these is really thinking about how we might actually incorporate really valuable information on what the predator is doing as a forager and how prey might be influencing their distribution abundance into our forecasts. Next slide. So a very common approach to trying to understand how we can predict species redistribution, as I've just illustrated with some of the work on species distribution models for albacore, is to relate past environmental conditions and species distributions, either from tracking or landings or abundance data, and then infer future distribution and abundance from predicted environmental changes. And what that might look like in the Eastern Pacific is essentially utilizing some of the rich data that's generated by regional ocean models to link that past distribution to future. Next slide. But a key step in the middle of these two connections between environment and distribution, of course, is understanding how interactions between predators and the species they forage on is going to moderate distribution and abundance. And that is a huge field of work that is utilizing large-scale food web models, often called end-to-end models, such as Atlantis or EcoSpace or EcoSim, that are trying to infuse these predictions with information on the feeding relationships between a focal predator and the prey base that they have available to them. These models are large, complex, and importantly, often very data hungry. Next step, slide. One of the key challenges to parameterizing models that use predator-prey information is also that many of the predators that are involved in the food web have complex foraging relationships and dynamics themselves. So as we have continued to study albacore tuna, what we've learned is that this particular predator alone consumes over 300 different prey types or species globally. They're distributed across ocean basins where they have access to a diverse set of prey fields. And as we look in more and more albacore stomachs and try to understand their diets, that taxonomic diversity continues to grow. Albacore in the past have been characterized as a bit of a generalist predator, in that they do have a very broad diet. But one of the key things that separates a generalist predator for one that is targeting particular prey types is the extent to which the diet that they consume reflects what is available to them in the environment. Albacore are, as they're distributed across oceans where the communities are changing in space and time, exposed to huge array of taxonomic diversity of prey. And so if we were to use this taxonomic diversity alone to try and understand what an albacore might do under future ocean conditions, we would be left floundering. Likewise, if we looked at just a few species that albacore consume at high rates in a particular time and space, that is likely not going to be predictive of what they might do when they're offered very different prey fields. Next slide. So the key lens that we have been bringing to this problem of trying to understand how the availability of forage or prey for top predators like albacore might moderate their redistribution and abundance changes under future climate states has been to put on our foraging ecology hats and to think about the process by which predators like an albacore or any other forager actually chooses their diet. So I want to take you a little bit away from albacore just for a couple of minutes to walk you through how this framework works. In particular, foragers like albacore or ourselves, we don't typically think of the diet choices that we're making as species. So for example, you or I might be going out to grab a snack or a meal, and we might be faced with a number of different options. Here, I'm just giving you the example of an apple or a pretzel. Sure, we might call them an apple or a pretzel, but when I'm deciding what I'm going to have, I'm actually looking at other attributes of these snacks. I am in particular thinking about their characteristics or traits and what I will be able to get in terms of benefit from them in relation to the cost. So I might characterize my apple as crunchy, sweet, gluten-free if that's important, high in vitamins, and easy on my wallet. Where I might look at the pretzel as soft and salty, with fewer vitamins but more calories and a little bit more costly. And if you observed my diet choices over many days or months or years, you might be able to start to build a picture of the characteristics or traits of the kinds of things that I typically choose, and you could use that information. Next slide. In particular, if we were to travel to a place such as another country where we had never been exposed to the particular foods that are available, the species or the types, we might be able to actually anticipate the kinds of diet choices that I or you might make from the characteristics of the things that we've eaten in the past. Essentially, the traits of our food would help to inform our foraging in new contexts. And this is exactly the lens that we're bringing to our studies on albacore tuna and other highly migratory ocean predators that are being faced with a redistribution of prey and new prey fields as climate change intensifies. Next slide. Trait-based analyses, as we'll refer to them for the rest of the talk, are on the rise across ecosystems. And in particular, this work has sort of burgeoned in terrestrial plant ecology. So many of the concepts and theories that we are bringing into this work have been tested in systems that are quite disparate to the one that we're going to be talking about today. But crucially, this trait-based lens allows us to have some process by which we can generate predictions about what might happen under novel conditions. And that's incredibly important, particularly as we think about global change forces, like a changing climate and how it's redistributing species, and therefore the prey that predators have access to. Next slide. What we have been doing in this project is thinking like a forager when it comes to the way in which albacore tuna consume their prey. We know that predators engage in a complex set of behaviors as they are seeking out prey in their environment, which is mediated by the abundance of prey types, their encounter with those prey in the environment, the extent to which they attack them, are able to capture them, and ultimately consume them. Typically, as ecologists, when we're trying to understand this process, we're just looking at the two ends. We're comparing what is consumed, often quantified through diet studies, and we may compare that to what's available as characterized by the abundance of prey types in the environment itself. And we might try to characterize diet selection by understanding the ratio or a comparison of those two properties, consumption to abundance or availability. But what we know from a huge wealth of foraging behavior research in other systems and also in open ocean systems is that traits of both the predator and the prey are very important in mediating the success of this process. In particular, properties such as the habitats occupied by the prey and their activity patterns modulate their ability to be encountered by predators, as well as their position in the water column. Physical and morphological properties, such as the presence of physical or chemical defenses, coloration, size, and shape, modulate the extent to which predators may attack or capture different prey. And in particular, shape and nutritional value will mediate the extent to which those prey items are benefiting the predator as they're consumed. And so what we have been able to do through our work is consider albacore tuna as a forager and the broad suites of prey that they have encountered in the past and may encounter in the future to understand the extent to which this variety of traits that we have strong hypotheses about influence patterns of diet selection and ultimately will help us to infer when and where albacore are consuming prey both now and into the future. Next slide. One of the key potential promises of a trait-based lens is that we may be able to use trait insights to understand how predators will utilize prey resources and therefore may track them into the future as the climate is changing. In particular, we're able to set up a couple of different hypotheses about just how useful a trait-based approach is, and that's really what we're trying to test in this project. If we think about the probability of prey traits in a predator's diet, and that's given by this sort of vertical or y-axis on the schematic graph here, and how that might vary across some environmental gradient, here that could be climate regime, cool or warm states, we could see that traits could be variably useful to us. In particular, the bottom trend here would be that the probability of traits in a predator diet is very random. And there really, in this case, is no predictive ability. We're not observing traits consistently, and we don't seem to be able to predict them across climate states. Similarly, at the top part of the graph, we might see that prey traits in the diet are very conserved, such that the same traits pop up no matter what the environmental conditions. And in this situation, traits would be incredibly valuable to us because we could predict what a predator might do, even independent of climate state. The middle scenario here, where a predator might switch between different prey types that are profitable in different environmental conditions, would also be useful to us, such that if we knew something about the traits that predators were interested in, and we knew something about the climate state or how that might change into the future, we could use traits to predict what the predator might do in conjunction with something like climate data. Next slide. The rest of the talk today is going to focus on our work with albacore tuna and their prey fields in the California Current large marine ecosystem specifically, because it provides a fantastic system under which to explore the potential for these trait-based approaches. In particular, the CCLME is a huge dynamic region that experiences both a north to south gradient in climate conditions, as well as oscillations over large timescales due to larger oceanic climate state forces. And so variable climate ocean conditions historically over time, and also across the region, across space, allow us to understand how albacore have been accessing prey in the future in relation to climate variability. And also, one of the key attributes of this system is the huge amount of observation and effort that scientifically that has gone into understanding this system. There is a wealth of rich biological data available both on albacore and their prey and many other predators in the system, as well as a number of large-scale collaborative efforts to understand and model the climate and what it will look like into the future. And so this is the system we're going to focus on for the rest of our seminar today, where we have been trying to test and gain insights into the utility of these traits-based tools. Next slide. Our ultimate objective of this work has been to develop and test a framework for integrating trait-based interactions into forecasts of species redistribution with albacore tuna as our focal lens, at least to start. The project has happened in 3 phases: a data synthesis phase where we have gathered insights on traits that are relevant to pelagic predator foraging, using those insights in combination with large-scale and long-time series data sets in the California Current to understand foraging dynamics under past climate states, and essentially test the utility of a trait-based lens in understanding those dynamics. And then following that hindcasting phase, looking to the future, in particular, trying to understand how prey fields for albacore, and therefore the predator that's accessing them, might be redistributed under future climate state scenarios in this system. So I'm now going to turn it over to Dr. Miram Gleiber to take us through some of our key findings across the various stages of this process and what our process has been.
Thank you, Stephanie. So now moving into our data synthesis phase. In order to facilitate trait-based work in pelagic systems, our group has assembled and made publicly available a trait database that we put online in 2022 and continue to update. We published a paper about it earlier this year in Scientific Data, which you can link to with the QR code. We include hundreds of species that are important for ocean predators globally, with a focus on prey for albacore and other top predators in the Northeast Pacific. We have over 30 unique traits that represent habitat and behavior, morphology, nutritional quality, and population status information. The traits are also life stage specific for adult, juvenile, and larval stages. These phylogenetic trees are showing the spread of the trait coverage for the trait forms across all 500 plus species in the database. I'm just going to briefly highlight some traits that we've been using in our trait-based analyses as key influencers on parts of the predation process. Habitat and vertical habitat, horizontal and vertical habitat positions, as well as activity patterns like diel and seasonal migration, impact encounter rates between a predator and a prey, since they physically need to overlap in space and time. Morphological traits for coloration, like countershading and silvering and transparency, impacts a predator's ability to recognize and attack their prey. We also collected morphometric ratios, which have been a useful way that are a continuous numerical way to examine body shape. And they've also been useful for species and stage-specific conversions from different length type values. For example, standard length or mantle length to total length. Nutritional quality impacts many aspects of the predation process. We include lipid content, protein content, and energy density, all standardized to wet weight. Of all the traits, nutritional content is the least resolved, and you can see the gaps in the phylotree represented by all the white spaces. For these taxa, we couldn't find any literature values, and this is an area of the database we're actively focusing on filling in data gaps. Traits were primarily collected from the literature based on keyword searches. We also used other online resources like FishBase and Tree of Life. These are incredible resources, but not ideally designed for trait extraction. We collected morphometric ratios by measuring multiple dimensions from images that we found online and confirmed to be the correct life stage and species. We've also been doing primary data collection through nutritional analyses, since nutritional content can vary across seasons, years, size, and many other variables. Alana Krug-McLeod, a master's student in our lab, is wrapping up a paper exploring variability between and among a range of species. And this is work we've been doing in collaboration with scientists at Oregon State University and Scripps. I'd like to acknowledge the students from our lab that really made this database the resource that it is today. Much of their work was done through the pandemic, and so while the lab group physically dispersed, these students were able to stay engaged in active research and have all moved on to wonderful next steps in their lives. So we've created this database not just for our own work, but to generally support trait-based ocean research. And we're excited to see that the database is starting to generate a global community of scientists. Some examples of how folks might use it include exploring trait associations, modeling energy flow in food webs, or applying traits to predator diet data. For example, in a recent study, Natasha Hardy, a former postdoc on the project, examined trait-based patterns of global albacore diets that included over 300 species. She applied 4 different habitat traits from the database that govern encounter rates between predators and prey. And when examined with a clustering algorithm, she found that for these broad, diverse diets of this global predator, they could be explained with just 7 different prey trait guilds that are shown here. And we're just really scratching the surface so far of what we can learn from this trait database, and open to collaboration if you see opportunities to use the database in your own work or add to it. So with our new trait database assembled, let's move into our hindcasting phase, where we explored the trait basis of albacore tuna foraging dynamics, focusing on the California current region. So we wanted to know if albacore show selective patterns of predation, looking at this from both a taxonomic and a trait-based perspective. To know what albacore are eating, we use the 3 modern albacore diet studies in this region that provides a 15-year time series from 2005 to 2019 across most regions of the California current. This includes an ongoing diet time series by NOAA Southwest Fisheries Science Center, where the stomachs are actually collected by the commercial fishery through a research nonprofit funded by the US West Coast albacore fishery, which I think is pretty cool. To get estimates of prey availability, we used annual NOAA pelagic trawl surveys towed to the depths of 30 to 45 meters, which is sampling within the foraging habitat of juvenile albacore. These surveys are the best available quantification of ocean species that might be available to albacore as prey, and used to develop ecosystem indicators as well as stock assessments. I want to take a moment and highlight the active role of the scientists that contributed this data that they had to our project. They provided not only support and feedback, but also contributed to writing and revisions as co-authors on the paper. These data sets are the result of years of work by these wonderful scientists, either identifying tuna gut contents under the microscope with incredible skill, or managing offshore field teams that are sampling with massive net systems in the unpredictable Pacific Ocean. And our team is excited that we were able to bring together these data sets with their generators to make the most of these key sampling efforts for collaborative analyses. In order to integrate the 3 surveys and the 3 diet data sets, we standardized taxonomic identifications, standardized counts as proportional abundances, and aggregated more rare taxa. We also assessed the suitability of the trawls for sampling albacore prey taxa by comparing the prevalence based on frequency of occurrence and the sizes that are sampled by both the trawls and the tuna. So now we have a time series of albacore diets, estimates of preyability in the system to go with it, and the traits to go with all of that. So this bar plot shows albacore diet mean proportional abundances of prey groups in each year and region. The notable absence of diet studies in the later years in the central and the south is due to the northward shift of albacore populations over this time series, with albacore rarely caught in these regions anymore. And we see a lot of interannual and regional variability in the diets, which is well known for this species. Now let's look at the trawl data representing availability in the system. We can see that the diets and the trawls do not track each other. Both have very different patterns of relative abundance across space and time. And we wanted to explore these relationships further to see if albacore are a more generalist, non-selective predator, or if there are selection patterns. So we used an electivity index to identify deviations from proportional consumption. The index we used is van der Ploeg and Scavia's relativized electivity, called E star, which ranges from -1 to +1, with 0 being neutral. So it's testing if the proportion of each prey type in the diet is as expected given all the prey types in the trawl composition. Warmer colors are positive E star values, identifying prey that are highly elected, meaning albacore are eating a prey type in greater relative proportions than in the trawls. Cooler colors indicate negative electivity, so lower relative proportions in the diet. And we evaluated electivity annually from both a taxonomic and a trait-based perspective. This heat map is showing the taxa-based results in the Northern California current, where we have the most years for comparison. Rows are taxa, and they're listed highest to lowest based on the overall mean electivity values across years. The colored circles just show instances where we couldn't calculate electivity, since that taxa is absent in that given year in either the diets or in the surveys. So we see that some prey are generally consumed at higher or lower proportions relative to the trawl composition, but there's a lot of interannual variability happening. And this tells us something different about albacore foraging than considering diet information alone. For comparison, I'm now listing on the taxa their rank order in terms of mean abundance in just the diet. The highest positive electivity values were actually for taxa that are not as abundant in the diet, but when examined in the scope of the available prey community, they're indicative of selection by albacore. For the sake of time, I'll just be showing results in this region, but know that in the south we see a similar pattern, just with slightly different taxa, and also highly variable across years. But tuna don't see taxa, they see traits. So let's put on our trait-based glasses and see what prey electivity looks like from a tuna's perspective. This heat map is showing the trait-based electivity in the Northern California current. We included 13 traits that can impact the predation process listed on the right. Electivity can only be calculated across groups that are mutually exclusive, so E star was calculated separately for each of these traits. And there's a lot going on here, but notice the consistent colors across rows, indicating consistent electivity patterns for traits across years. In fact, 8 of the 13 traits have trait forms we examined that are consistently highly elected for by albacore, with the alternative trait form having strong negative electivity. And just for fun, let's also take a look at the south. The pattern's actually the same as the north. The same trait forms are positively elected across years, and these trait forms were also consistent outcomes of a multivariate analysis that we conducted, but I don't have time to present today. These density plots are showing the relative amounts of variance in E star, so electivity, across years from all taxa compared to all traits in each region. And we see that the interannual variability in taxonomic albacore prey electivity, that's shown in blue, is ultimately minimized through a traits-based perspective, which is shown in yellow. So by examining albacore diets in the context of available prey, we found that electivity highlights different indicators of predator-prey interaction strength than examining diets alone. But the interactions still remain taxonomically complex across space and time. However, when we examine selection patterns through a traits-based lens, we see that in this predator with variable diverse prey, the same traits are selected across years and regions with vastly different environmental states. We recently just published this in Ecological Indicators, and Natasha and I were co-lead authors on this work, and you can link to it with the QR code here. So at this point, we've used the trait database to examine the foraging dynamics of albacore under past climate states in the California current, finding that for these highly variable complex diets, albacore tuna, in albacore tuna, we see the traits are not only useful in explaining feeding patterns, but actually do simplify the spatial and temporal differences. And figuring out where these preferred prey go will likely structure where the predators go. So these consistent trait-based selection patterns can now provide a new perspective on albacore feeding that we can use to help forecast changes in albacore distributions into the future. So based on these findings, we're now asking when and where are the most preferred prey for albacore across time and space. We're exploring this question by scoring taxa based on the number of preferred traits they have. So each taxa is represented by what our group is now calling a yumminess score. For example, anchovies and sardines have the highest yumminess score. So out of the traits considered, they have the most traits preferred by albacore. We can apply this to trawl data to calculate a yumminess index by weighting each taxa's score by the abundance and summing this across all the taxa in the trawl. We applied this to the time series of forage community data in the California current to identify hotspots of highly suitable prey for albacore, indicated by the warmer colors. These maps are showing an example of how this looks before, during, and after the marine heat wave from 2014 to 2016, and we see an apparent increase in community yumminess in these warm years. In fact, when we look at annual anomalies of trawl yumminess in each region across the time series, it appears that the shift in the forage community across regions in these anomalously warm years has resulted in more highly suitable prey for albacore. So now we're exploring the predictive ability of this traits-based approach to project when and where the most yummy prey for albacore will be into the future. We're using environmental conditions associated with all of these trawls to build distribution models of preferred prey for albacore. Then applying the model to future projection data in the region to predict the future distributions of preferred albacore prey. I want to take a moment and quickly highlight the amazing resource that we have in the California current for studying environmental conditions of this system into the future. How this works is 3 of the many global Earth system model projections are downscaled to the California current region using regional scale models. This provides a high-resolution hindcast and projections of conditions, including temperature, wind stress, biogeochemistry like oxygen and nitrate that also provide chlorophyll A as a measure of biological productivity. Here we can see examples of the high-resolution data for sea surface temperature historically, and then the 3 different projections that show change in sea surface temperature by the end of this century. So this is the wealth of information we have in the California current to model into the future. And before you get too excited, we haven't done this quite yet. We're still making final decisions on the distribution model. So for the distribution model, we're using boosted regression trees to predict yumminess index from trawls using sea surface temperature, surface chlorophyll A, beauty, which is an upwelling index, and habitat compression index, which is a regional scale indicator used for informing ocean management. It describes the extent of cool surface water conditions off the California current. The data product we're using is the data assimilative reanalysis of the ROMs, which integrates the regional model hindcast with direct measurements from satellites and in situ instruments. So by using this ROMs product at this step, it will help reduce the bias later when we're applying the projection data. We used prior conditions based on a January through June average, which represents preconditioning of the forage community prior to summer and perform better than other models that we've tested with present or other ranges of prior conditions. The models were trained on 80% of the data and tested on the 20% withheld at random. So we've seen that sea surface temperature had the strongest influence on predicted prey yumminess, with the most preferred prey for albacore following warmer early season conditions. Chlorophyll A was also important, with yummier prey for albacore following low or high productivity, but not mid-range prior to summer. For habitat compression index, higher prey yumminess for albacore was predicted following moderate HCI conditions, but lowest when the cool upwelled water was either more compressed or more expanded along the coastline in the early season. Interesting to see these opposing U-shaped patterns for chlorophyll and HCI, possibly highlighting groups of foragers with different responses. And upwelling had the lowest influence overall, but was still important, with preferred prey following higher upwelling conditions. Overall, the best model has a CV deviance on the test data of 0.27. We're excited to see these interesting results that are staying consistent as we make some final adjustments with the data. So what's next? We'll be applying the boosted regression tree model for yumminess to the projected ROMS data to forecast prey fields into the future. Here we can see how key environmental predictors of preferred albacore prey, like sea surface temperature and chlorophyll, are projected to change by the end of the century in this region. So you can imagine a similar type of spatial temporal prediction of yumminess into the future. And from this, we can answer questions such as, where will hotspots of preferred prey be into the future? Or how variable are prey fields likely to be in location and quality? We're also delving further into prey quality and estimating the nutritional quality of trawls to look at the energy or lipid contents of the forage community across the region, with the database as a resource to apply length and weight specific regressions for nutritional metrics for a range of key species. And the goal here is to forecast spatial temporal trends in forage quality into the future. Now I'm going to turn things back over to Stephanie as she wraps up with our vision of where this project goes into the future.
Thanks, Miram. I want to take a minute just to highlight some of our thoughts on future directions and some of the framing of this project and what we might see next. As Miram has outlined, this journey we've been on the last few years has really highlighted to us that trait-based frameworks may prove very useful in understanding what predators have done in the past under variable climate conditions, and therefore what they might do in the future. And as we've generated those trait-based insights in this project, we have a keen interest in seeing those insights being used and adapted into other ways of understanding potential change. One of the major future directions that we're hoping to continue on with this project is testing the performance of these trait insights and frameworks, particularly the multivariate traits lens that we've been able to take within existing modeling frameworks, both within models such as trait-based species distribution models, but also larger, more complex models that integrate other modeling types. Here's just depicted a schematic of, for example, the end-to-end ecosystem model Atlantis. The middle dark purple boxes are the predator-prey linkages that connect the key parts of the ecosystem that integrate climate and oceanographic change and percolate those changes up through into what is available, for example, to fisheries at the coast. Next slide. We also see a huge potential to use both the data we've already synthesized and the insights we've generated in the traits database, as well as the California current information that we have worked with partners to synthesize and clean, to think about extending this prey traitscape framework to other pelagic predators, perhaps both in the California current large marine ecosystem, where species of prey are shared widely across a number of pelagic predators. The schematic here is just a figure that comes to us from a predator diet data set that has been assembled by colleagues at NOAA, integrating information on the stomach contents of predators in the marine environment in the CCLME over many decades and many taxa. And so our hope is that the resources we've assembled and the insights we've gained through this project can help others who are trying to understand the dynamics of predator populations and their distributions from other taxa in their work. Next slide. I also want to speak just briefly to the bigger picture of this project, because the project that we have been working on, funded by Lenfest, has benefited enormously through collaboration and dialogue with many colleagues with NOAA Fisheries at both the Southwest and Northwest Fisheries Science Centers, particularly through the Future Seas Project initiative. Future Seas started, I think, at least 5 years ago now, with an initiative that was looking to provide a physics to fisheries management strategy evaluation for the California current large marine ecosystem. And to me, it's one of the most amazing examples of bringing people together to share insights, lessons, and resources to think about how different streams of science can inform how the system is changing and what it means for resource availability and resource use. In particular, we have benefited through dialogue and collaboration as many other types of ecological models have been developed for coastal species within the CCLME, including albacore tuna, including individual-based population dynamics and species distribution models, as well as economic and social models that have looked at landings and other analyses. And the outputs of those efforts by colleagues have provided insights that have been directly involved in conversations with stakeholders as well as managers who are thinking about how to ensure the sustainability and access to resources, fisheries resources on the coast. And so we're incredibly grateful for those partners and that partnership. And also that partnership has allowed us to think about how we both frame and conduct the project that's focused on the trait lens, but also given us a wealth of ideas for how these data could be used in subsequent steps to think about how we can inform management through channels that already exist in the management framework. Next slide. I want to also say that we're very eager to continue the conversation. This project has been ongoing for the last 5 years, and we're just in the final phases of it. However, we see that the work has, we really think, got to continue. There's so much to do and so much we are now just learning, and we want to make that work as relevant as possible for the questions that folks like you in the audience have. So in particular, if you want more information about what we've done and some of the findings that we've shared in summary today, we'd welcome hearing from you. If you have feedback or insights on how some of this could be useful or how you'd like to see some of this work proceed going forward. And equally, if you're interested in potentially collaborating or using some of the output, we would welcome follow-up conversation and dialogue with you and would love to hear from you. Next step. In summary, I want to thank everyone here for their time in attending this webinar. In particular, I want to thank all of our collaborators and colleagues for their work on this project. And I also want to thank the Lenfest Ocean Program for their immense support in making this work possible and being a wonderful team to work with in achieving all of the outcomes that we'd set out at the beginning of this several years ago. I'd also invite you, if you're interested in exploring the pelagic species trait database that we mentioned and described in the webinar today, you can use this QR code here, or you're welcome to contact us by email. Thank you again.
Fantastic. Thank you both for such a great presentation. This project is so interesting every single time I hear about it. People are welcome to submit questions in the question and answer box. We have a few minutes, and if people need to drop off right at the top of the hour, please feel free to do that. But if there are questions, I would love to be able to get to them while we have Stephanie and Miram with us today. I will say that all of the pages or all of the publications that were mentioned in this presentation are on that resources page link that we mentioned earlier in the chat. We'll also share this with the webinar recording. We do have one comment in the chat that just says, fantastic work, fantastic presentation, really appreciate the links to the literature and they're a big fan of the Yumminess Index, as are we. Very excited to see where this work goes in the future. I'm not seeing any questions in the chat, but I do have one question for both of you while we're here. I know that this is, a relatively new concept, and I'm just curious to hear from you what it would take to take this from theory to practice, in terms of next steps for this project.
Yeah, Kayla, I think we're right on the cusp of the practice part in that we have taken it from this essentially hypotheses we had about what, for example, albacore tuna might do. We've gathered what we feel is fairly strong evidence of what they do, and we're now looking to the future to try and understand how, for example, the prey resources they rely on are going to be potentially looking into the coming decades. And so I think that's the really exciting part, is we're right at the point. Unfortunately, for the timing of the webinar, we're not quite there yet, but we're right there at being able to look ahead and provide ideally that information in a visual way that would be one more input into considering, for example, how a predator like albacore will be distributed in the Eastern Pacific in decades to come and what that might mean for accessing that resource. Miram, I wonder if you have other things to add.
Absolutely. I would agree with all that. I think it's interesting as we look towards the future and you think about individual species, and it can be often hard to model all of those populations. But when we think of this as a big aggregate, a composite of traits, we're really excited to see where we can predict those showing up and those abundance of those preferred species into the future, thinking about key issues like, the US-Canada treaty negotiations that are going on and the information that it can provide to understand where key prey for this, fisheries important species are moving.
Definitely. We did get another question in the chat. How many of the variables in your trait database do you think would be useful for explaining or predicting prey digestion rates? For example, jellyfish. They won't show up in many diets because they get digested very quickly.
Yeah, thanks for the question, Mark. I'll start off and then Miram probably will have something to add. But I think one of the really exciting things about having this database now is that it allows us to test some of our assumptions and questions we might have about the patterns we're seeing in the data. In particular, as you mentioned, digestion rates are such an important consideration when we think about how it might impact what we observe to be the types of prey that are utilized by a predator in terms of skewing our observations there. I'm really excited about thinking about the intersection between, for example, body plans and forms and morphology, nutritional profiles of these species, and then what we do see in terms of their use by predators, because I would suspect that there's going to be, for example, quite a bit of correlation among some of these traits. And we might be able to use information, for example, on nutritional profile to look at something like digestion rate and also profitability at the same time.
I would also add to that specifically for this question, which is a really great question. As Stephanie mentioned, we have this nutritional quality component of the database, which includes lipid content, protein content, energy, and a lot of that information, when you think about it across species globally, could get at some of these questions about prey digestion. There's also like a cryptic trait in there of water content, which is something we've cataloged along with all of these nutritional metrics. And so something like that could also be really useful for this type of question.
Great. Wonderful. And I think that's all we have time for today. But if you would like to reach out with other questions, please feel free to reach out to Stephanie or Miram, others on the research team, and us at Lenfest. We'd be happy to follow up. And with that, we'll go ahead and end the webinar. And I'll just end by saying thank you so much, Stephanie and Miram, for all of your hard work. And this was a great presentation. So thank you.
Thank you.
Thanks,
Bye,
everyone.
Thanks.